Lecture Notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610456763. 3. Enzymes. Enzymes are proteins that help speed up chemical reactions in our bodies. Enzymes are essential for digestion, liver function and much more. Too much or too little of a certain enzyme can cause health problems. Enzymes in our blood can also help healthcare providers check for injuries and diseases. 3.1 Mode of action of enzymes. Enzymes are biological catalysts, reducing the activation energy of reactions. They are composed of globular proteins with a specific tertiary structure. Increasing temperature increases the kinetic energy of enzyme and substrate molecules, with high temperature causing the enzyme to become irreversibly denatured. Enzymes operate in narrow pH range, with changing pH around the enzyme disrupting the active site. Increasing enzyme concentration causes a linear increase in the rate of an enzyme-catalyzed reaction, whereas increasing substrate concentration also increases rate, but only until a maximum rate is reached. Competitive inhibitors bind to the enzyme's active site, whereas non-competitive inhibitors bind elsewhere on the enzyme. Enzymes are used in industry because they allow reactions to occur at normal temperatures and pressures. For example, clarification of fruit juice occurs through the pectinase enzyme. Enzymes control metabolic reactions in the body and the rate at which they occur. Metabolic reactions can be catabolic, breakdown reactions, which release energy overall, or anabolic, building up or synthesizing reactions, requiring energy overall. Characteristics of enzymes. Biological catalysts, made up of globular proteins with a specific tertiary structure. They reduce the activation energy of reactions, which is the energy required for a specific reaction to occur. They speed up and slow down metabolic reactions but are never used up in the reaction. Enzymes, like all catalysts, do not make reactions occur that would not otherwise happen and do not change the volume of product finally produced. The active site is a depression in an enzyme's tertiary structure and is the site of the enzyme-catalyzed reaction. The shape of the active site allows only specific substrate. Lecture Notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610456764. Molecules to enter, which results in enzyme specificity, with enzymes unique to a specific reaction. The enzyme and substrate are said to form a steric relationship. There are two theories about the action of enzymes. The rate of enzyme-catalyzed reactions will increase with increasing temperature, up to an optimum rate, the temperature producing the highest reaction rate. Above this optimum, further temperature increase decreases the rate of reaction. Increasing temperature increases the enzyme and substrate molecules' kinetic energy. The faster-moving molecules collide more often, forming more enzyme-substrate complexes. Temperatures above optimum cause increasing enzyme molecule vibration, breaking down internal bonds and destroying the active site. The enzyme becomes irreversibly denatured. Enzymes are very sensitive to pH. Individual enzymes operate in a narrow pH range and either side of this optimum irreversible denaturation will occur. Optimum pH for most enzymes is 7, neutral, although some digestive enzymes, e.g. pepsin, have their optimum rate in acidic conditions. Changing pH causes changes in the change of an enzyme's amino acid components. This alters attraction and repulsion forces within the enzyme, disrupting the shape of the active site. The rate of an enzyme-catalyzed reaction is also affected by concentration of both enzyme and substrate. Enzyme concentration. Increasing enzyme concentration causes a linear increase in rate, provided the substrate concentration is not limiting. Increasing enzyme concentration increases the number of He active sites available to catalyze the reaction. Substrate concentration increasing. Substrate concentration maximum rate is reached, plateau. Maximum rate is due to all available active sites being occupied by substrate molecules. Increasing substrate molecules will therefore not increase the rate. Enzymes are widely used in industry because they can accomplish reactions at normal temperatures and pressures, saving the cost of expensive, energy-demanding high temperatures and pressures. The enzymes may be extracted from cells and purified or used within cells. Enzyme industry application. Pectinase fruit juice manufacture. Clarification of fruit juice. Proteases biological detergent manufacture used for pre-soaking and direct stain removal. Problems may arise in using enzymes for detergents as people develop allergic reactions. This can be overcome by preventing the enzyme coming into direct contact with people, encapsulation techniques. Enzymes are globular proteins with a precise, but delicate, three-dimensional shape maintained by ionic and hydrogen bonds. During a reaction, the substrate fits into a region on the enzyme surface called the active site. Enzymes are specific. Each enzyme catalyses one particular reaction. Enzymes speed up reactions by lowering the activation energy needed to get the reaction started. Between 4 OC and 40 OC, the rate of an enzyme-controlled reaction increases for every 10 OC to double in temperature. Increase in temperature beyond 40 OC usually denatures the enzymes. Activity is lost when its three-dimensional structure is destroyed. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455765. Most enzymes have an optimum pH. For intercellular enzymes this is usually about 7.35. Extracellular enzymes, such as found in digestive juices, may have optimums of extreme pH values. The rate of an enzyme-controlled reaction is limited by the supply of substrate, enzyme or the enzyme cofactor. Inhibitors are substances that slow down or stop enzyme activity. They may be reversible or non-reversible. Reversible inhibitors may be competitive or non-competitive. Competitive inhibitors tend to be similar in structure to the substrate and compete for the active site. Non-competitive inhibitors do not bind to the active site itself but alter the shape of the enzyme so that the active site is no longer functional. Enzymes consist of globular proteins. Enzymes act as catalysts by lowering activation energy through the formation of enzyme-substrate complexes. Temperature, pH, concentration enzyme and concentration of substrate affect the rate of enzyme-controlled reactions. Competitive and non-competitive inhibitors decrease the rate of enzyme-controlled reaction. 3.1-to-1 enzymes, globular proteins. Fibrous proteins often join to form large fibrils whose function is to provide strength or produce movement. Globular proteins, including enzymes, antibodies, and some hormones, are usually individual molecules with a chemical function. 3.1-to-2 Mode of action of enzymes. Lock and key hypothesis, induced fit hypothesis. When a substrate enters an enzyme's active site, the enzyme and substrate form a steric relationship. In the lock and key hypothesis, substrate shape is complementary to the shape of the active site. Lock and key, substrate shape is directly complementary to the shape of the active site, with the enzyme acting as the lock and the substrate molecules the key. There are two models of enzyme action, the lock and key, and induced fit models. Induced fit, not all enzymes have a permanent active site. In some, one develops as substrate molecules come close. A small change occurs in the enzyme structure to form a specific active site. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455766. 3.1-to-3 Investigation of the progress of enzymes catalyzed reaction. The course of enzyme reaction can be followed by measuring the rate at which a product is formed or the rate at which a substrate disappears. A progress curve, with time on the x-axis, can be plotted. The curve is steepest at the beginning of the reaction, when substrate concentration is at its highest. This rate is called the initial rate of reaction. 3.1-4 The use of a colorimeter for measuring the progress of enzymes. Catalyzed reaction colorimeter is a light-sensitive device that measures the absorbance of transmission of certain wavelengths of light by a solution. A colorimeter can be used to investigate the rate of a reaction that involves color changes. 3.2 Factors that affect enzyme action. Enzyme activity can be affected by a variety of factors, such as temperature, pH, and concentration. Enzymes work best within specific temperature and pH ranges, and suboptimal conditions can cause an enzyme to lose its ability to bind to a substrate. 3.2-1 Temperature affects the rate of an enzyme-controlled reaction in two ways. As the temperature decreases, the kinetic energy of the substrate and enzyme molecules decreases, so they move slower. Thus, they do not collide as frequently with each other, and this causes the rate of the reaction to be slowed. 2. As the temperature increases, the substrate and enzyme molecules have more kinetic energy for them to move faster. Thus, they collide more frequently with each other, and this causes the rate of the reaction to increase. However, with increasing temperature, the atoms of the enzyme molecules vibrate more until the hydrogen bonds and other forces holding the molecules start to break. The protein structure of the enzyme molecules is altered such that the substrate no longer fits into the active site. The enzyme is then deemed no longer catalytically functional. This occurs at about 60 OC and the enzyme is denatured. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457767. pH. Using buffer solutions, every enzyme functions best over a particular range. The maximum rate of reaction will occur at the optimum pH for each enzyme. If a deviation from this optimum pH occurs, the rate of reaction will decrease due to reduced catalytic activity of the enzyme. The deviation in pH can also effectively denature enzymes by altering the enzyme shape and subsequently its active site. This is because pH is a measure of the concentration of hydrogen ions. The change in concentration of hydrogen ions present may alter the ionic charges in the enzyme and break the hydrogen bonds, maintaining the shape of the enzyme molecules. Enzyme concentration. The rate of an enzyme-catalyzed reaction is directly proportional to the enzyme concentration, provided that there is a high concentration of substrate molecules and other conditions for optimum function are maintained. This is because the active site of an enzyme may be reused. Therefore, the more enzymes are present, the more active sites will be available for the substrate molecules. Thus, the turnover number, the number of substrate molecules that an enzyme can act upon in a given time, will be greater and the rate of reaction will increase. Substrate concentration. The rate of an enzyme-catalyzed reaction increases as the substrate concentration increases until a point where the rate does not increase further. This is because the active sites of the enzyme molecules are not all used up when the substrate concentration is low. However, if the substrate concentration is increased, more active sites are used up and the rate of reaction becomes faster. When all the active sites are being used, the rate of reaction cannot increase further as the amount of enzyme becomes the limiting factor. Inhibitor concentration. Inhibitors are substances that prevent normal enzyme activity. When they are added to a mixture of enzyme and substrate, the rate of reaction is reduced. Inhibitors combine with the enzyme to form an enzyme inhibitor complex, so the enzyme cannot combine with any substrate molecules. Competitive inhibitors compete with the substrate for the active site. The level of inhibition is dependent upon the relative concentrations of substrate and inhibitor, with increasing inhibitor concentration increasing the level of inhibition. This type of inhibition is reversible. Non-competitive inhibitors bind to the enzyme, but not at the active site. The level of inhibition depends only on the concentration of the inhibitor, not the substrate, because the inhibitor has a higher affinity for the enzyme than the substrate. This type of inhibition is irreversible. 3.2 to 2 Michaelis-Menten constant, Km. The substrate concentration at which an enzyme works at half its maximum rate, 1/2 Vmax, used as a measure of the efficiency of an enzyme. The lower the value of Km, the more efficient the enzyme. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457768. Vmax. The theoretical maximum rate of an enzyme-controlled reaction, obtained when all the active sites of the enzyme are occupied. 3.2 to 3 Reversible non-competitive inhibitors. These bind at a site elsewhere on the enzyme, causing a change in shape of the active site. Competitive inhibitors. Competitive inhibitors compete with the substrate for the active sites of enzyme molecules. While it remains bound to the active site, it prevents substrate molecules from occupying them, so the rate of reaction is reached. The same quantity of product is formed because the substrate can continue to use other enzyme molecules which are unaffected by the inhibitor. However, it would take longer to make the products. If the concentration of the substrate increases, less inhibition occurs. This is because the greater the proportion of substrate molecule, the greater their chance of finding active sites. An example is molecule acid, which competes with succinate for the active sites of the enzyme, succinic dehydrogenase, in the Krebs cycle. Although the rate of enzyme-controlled reactions may be decreased by the presence of inhibitors, this type of inhibition can be reversed by removing the competitive inhibitor. The effect of reversible inhibitors is temporary and causes no permanent damage to the enzyme because the association between the enzyme and inhibitor is a loose one. The removal of the inhibitor restores the activity of the enzyme to normal. Non-competitive inhibitors. Non-competitive inhibitors do not attach themselves to the active site of the enzyme, but elsewhere on the enzyme molecule. They alter the shape of the enzyme molecule in such a way that the active site can no longer properly accommodate the substrate. As the substrate and inhibitor molecules do not compete for the same sites, any increase in substrate concentration will not reduce the effect of the inhibitor. An example is cyanide, which attaches itself to the copper prosthetic group of the enzyme, cytochrome oxidase, thereby inhibiting respiration. Those inhibitors that leave the enzyme permanently damaged and unable to carry out its catalytic function are known as non-reversible inhibitors. For example, toxins, such as parathion and sarin gas, inhibit enzymes involved in the nerve activity in heavy metals, such as mercury and silver, cause disulfide bonds to break. As those bonds help maintain the enzyme shape, the enzyme molecule structure becomes irreversibly altered when the bonds are broken. Thus, there is a permanent loss of its catalytic properties. 3.2 to 4 Enzymes can be immobilized, for example, by trapping them in jelly, alginate, beads. This is commercially useful because the enzyme can be reused and the product is separate from, uncontaminated by, the enzyme. Immobilization often makes enzymes more stable. Uses of immobilized enzymes. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457769. Immobilization is the process of attaching an enzyme to an inert, solid support. These enzymes are less likely to be denatured pH or temperature. However, this technique cannot be used for complex reaction pathways. Compared to free enzymes in solution, immobilized enzymes are more robust and more resistant to environmental changes. More importantly, the heterogeneity of the immobilized enzyme systems allows an easy recovery of both enzymes and products, multiple reuse of enzymes, continuous operation of enzymatic processes, rapid termination of reactions, and greater variety of bioreactor designs. Immobilized enzymes can be separated easily from reactants and products can thus give a higher degree of control. They are also more stable. Immobilization is the process of attaching or trapping an enzyme to an inert, solid support, e.g. agar gels or cellulose. Advantages. Disadvantages. Enzymes can be reused, saving the cost of continual production. Enzymes are less likely to be denatured by pH or temperature. Initial cost of immobilization is high. Technique cannot be used for complex reaction pathways. Enzyme technology. Extracellular enzymes are secreted to function outside the organism's cell. Intracellular enzymes function inside the cell. The commercial production of enzymes involves the growth of large numbers of microorganisms using specific media and aseptic conditions. The commercial production of enzymes involves the isolation and purification of the enzyme product by downstream processing. Enzymes may be used as analytical reagents because of their high sensitivity and specificity. Industrial processes require a high degree of thermostability. Industrial uses of enzyme technology. Industry enzyme use dairy industry lipases. Blue cheese ripened by extracellular lipases from mold, e.g. Penicillium roquefortii. Brewing industry amyloglycosidase breaks down sugars in production of low-calorie beer. Baking industry proteases lowers protein content of flour for biscuit production. Agricultural industry lignases. Most wood waste is not very useful as it contains lignocellulose and few organisms can utilize this. Lignases from Sportricum pulverulentum make cellulose available for animal feed. Leather industry. Trypsin removes hair and excess tissue from hides and skin to make the leather more pliable. Trypsin used to be obtained from dung. Medical. Trypsin dissolves blood clots and cleans wounds. Biological washing powders. Mostly bacterial extracellular proteases remove organic stains from washing. Factory workers may become allergic. Immobilization of enzymes overcomes this. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 030046104570. Keywords: activation energy, active site, amylase, catalyst, colorimeter, competitive inhibitor, enzyme, enzyme/substrate complex, extracellular enzyme, extracellular reaction, immobilized enzymes, induced fit hypothesis, intracellular enzyme, intracellular reaction, lock and key hypothesis, Michaelis-Menten constant, Km, non-competitive inhibitor. Substrate specificity, Vmax. Chapter Glossary. Activation energy is the amount of energy needed for a reaction to happen. Active site is a specific region on an enzyme where the substrate binds and the reaction takes place. Amylase is an enzyme that catalyses the extracellular breakdown of starch. Catalase is an enzyme that catalyses the intracellular breakdown of hydrogen peroxide into oxygen and water. Catalyst is a substance that increases the rate of reaction, often by offering an alternative reaction pathway with a lower activation energy. It is unchanged at the end of the reaction. Competitive inhibitor is a molecule which binds to the active site of an enzyme and prevents the substrate from binding. Enzyme is a globular protein molecule that acts as a biological catalyst and increases the rate of biochemical reactions. Some enzymes work intracellularly and others work extracellularly. Enzyme/substrate complex is the temporary complex formed when the substrate binds to the active site of the enzyme. Extracellular enzyme is an enzyme that is secreted by cells and functions outside of cells, e.g. amylase. Extracellular reaction is a reaction that occurs outside of cells, e.g. in the tissue fluid. Immobilized enzymes are enzymes which are attached to an inert, insoluble material over which the substrate passes and the reaction takes place. Induced fit hypothesis is a model of enzyme action that describes how enzymes undergo subtle conformational changes which exert a strain on bonds in the substrate. Intracellular enzyme is an enzyme that acts within cells, e.g. catalase. Intracellular reaction is a reaction that occurs within cells. Lock and key hypothesis is a model of enzyme action that describes how the enzyme will only fit a substrate that has the correct complementary shape to the active site. Michaelis-Menten constant, Km, is a value which indicates how strong the affinity between an enzyme and its substrate is. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457771. Non-competitive inhibitor is an inhibitor which binds to a part of an enzyme which is not the active site, an allosteric site, and prevents the enzyme from functioning. Non-competitive inhibitor is the ability of an enzyme to catalyze only a specific reaction or set of reactions which have substrates complementary to the active site of the enzyme. Vmax is the maximum reaction rate. Short MCQs with right answers. 1. CYP3A4 is an important enzyme in the human digestive system where it is needed to break down a range of different toxins. The activity of CYP3A4 has been shown to be reduced by substances called furanocoumarins. Furanocoumarins are found in some fruits, and so dangerous concentrations of toxins may develop in the human digestive system when fruits containing furanocoumarins are eaten. From the information provided, what can be concluded about molecules of the enzyme CYP3A4? They lower the activation energy of the toxin breakdown reactions. 2. A fixed volume and concentration of substrate and enzyme were mixed. All other variables were kept constant. The enzyme-catalyzed reaction was left until it was complete. Which graph shows how the rate of reaction changes with time? 3. Some animals produce antimicrobial proteins which protect them from pathogens. These proteins could be used to kill human pathogens, however when used as a medicine they are broken down by protein-digesting enzymes. Replacing one of the amino acids found in the protein with an amino acid that had been synthesized in the laboratory resulted in a modified protein that was not broken down. What could explain why this modified protein was not broken down by the protein-digesting enzymes? 1. The modified protein has a different tertiary structure to the original protein. 2. The modified protein is not complementary in shape to the enzyme's active site. 3. The modified protein is unable to induce a fit with the protein-digesting enzyme. 4. A student investigated the effect of substrate concentration on the rate of an enzyme-catalyzed reaction. A graph was plotted to show the relationship between these two variables. The student was asked to take readings from the graph that could be used to determine the Michaelis-Menten constant, Km, for this enzyme. J, K, L and M show points read from the graphs which the student could use to determine the value of kilometer. Which two readings must the student use to determine the value of Km? Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457772. 5. The end product of a metabolic pathway can act as a competitive inhibitor. This is called end product inhibition and allows a cell to control a metabolic pathway. The diagram shows a metabolic pathway where the end product could act as an inhibitor of enzyme W. What would be the effect if enzyme Z was inhibited by the end product instead of enzyme W? 6. Which statement describes an example of an extracellular enzyme? Amylase in saliva is an enzyme that catalyses the breakdown of starch in the mouth. 7. Which row is correct for enzymes that catalyze reactions using the lock and key hypothesis? Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457773. 8. A scientist investigated the rate of breakdown of hydrogen peroxide. 4 experiments were carried out using different mixtures. Subjects Substrate only, substrate plus enzyme, substrate plus enzyme plus competitive inhibitor, substrate plus enzyme plus non-competitive inhibitor. The results are sketched in the graph. 9. Which row shows the correct lines for 2 of the experimental mixtures? 10. Which enzyme is extracellular? Amylase in saliva is an enzyme that catalyses the breakdown of starch in the mouth. 11. Protease enzymes are found in many locations inside and outside the cells. Which of the given locations are sites of intracellular hydrolysis? 1. Lysosomes. 2. At a telophase spindle only 12. Some extracellular protease enzymes are synthesized in an inactive form called zymogen. Zymogen is converted to the active protease in the Golgi body. What is the purpose of producing zymogen before converting it to the extracellular protease? To avoid unwanted digestion of proteins inside the cell. 13. Which statement about the active site of an enzyme is correct? It is determined by the primary structure of the enzyme. 14. What is the effect of an enzyme-catalyzed reaction? Decreases the activation energy and has no effect on the energy yield. 15. Which words from the table correctly complete the paragraph about enzymes? When the pH of an environment is decreased below the optimum pH of an enzyme, hydrogen and ionic bonds between adjacent R groups, holding the tertiary structure, are disrupted. 16. Which of these statements describe the action of an extracellular enzyme? Digestion of macromolecules in the lumen of the small intestine. 17. What determines the specificity of an enzyme? The bonding between R groups of the polypeptide. 18. HIV-1 protease is an enzyme produced by the HIV virus. 2 identical chains of 99 amino acids form the enzyme. In each chain, amino acids 25, 26 and 27 in the sequence form part of the active site. Which orders of protein structure controlled the shape of the active sites? Primary, secondary, tertiary and quaternary. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457774. 19. Which statements are true about the optimum temperature of all enzymes? It is temperature at which the enzymes work best. 20. Which of the statements describe some roles of enzymes? 1. Catalyze the breakdown of larger molecules into smaller ones. 2. Lower the activation energy required to start a reaction. 21. Which statements are correct for all enzymes? 1. They are globular proteins. 2. They can be inhibited by competitive inhibitors. 22. The enzyme lysozyme secreted from tear glands forms deposits on contact lenses. Which ingredient would be effective in a contact lens cleaner for removing these deposits? Proteases. 23. When investigating the rate of reaction of the enzyme lipase on the hydrolysis of triglycerides, the pH must be maintained at an optimum to prevent the lipase denaturing. What is the reason for this? The products of hydrolysis decrease the pH. 24. What may fish living in ponds survive temperatures below 0 OC in winter? The ice on the pond surface insulates the water in the pond. 25. What is the role of enzymes in metabolism? To lower the activation energy required to start a reaction. 26. What is the effect of an enzyme in an enzyme-catalyzed reaction? Decreases the activation energy and has no effect on the energy yield. 27. Glucose in urine can be detected using a biochemical test. When the end of a test strip, which is impregnated with the enzyme glucose oxidase, is dipped into urine, the development of a blue color indicates that glucose is present. This is a reliable test that people with diabetes can carry out at home. Which feature of the enzyme makes this test so reliable? It is specific. 28. Which statements describe some enzyme actions? 1. Enzymes hold reacting molecules so that their reactive groups are close together. 2. In an enzyme-catalyzed reaction, more molecules have sufficient energy to react than in the absence of the enzyme. 3. Reactions catalyzed by enzymes take place at a lower temperature than they would without the enzyme. 29. Which levels of protein structure would always be changed if an enzyme works by the induced fit hypothesis? Primary, not changed; secondary, not changed; tertiary, always changed; and quaternary, not changed. 30. Which statement explains how the substrate is able to enter the active site of the enzyme? When the enzyme/substrate complex forms, the tertiary structure of the enzyme changes. 31. Which statements are correct for the induced fit hypothesis? 1. The shape of the active site changes to enable the substrate to bind. 2. Temporary bonds hold the substrate in the active site. 3. More enzyme-substrate complexes are formed at the optimum temperature. 32. What could be used to calculate the rate of an enzyme-catalyzed reaction? 1. The temperature of product. 2. The disappearance of substrate. 33. Which statement is only true for the induced fit theory of enzyme action? The enzyme changes shape in the presence of substrate. 34. The graph shows the concentration of one of the substances which is involved in an enzyme-catalyzed reaction. Which substance is shown by line X, parallel to timeline on the graph? Enzyme. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457775. 35. Which statements describe some enzymes' reactions? 1. Enzymes hold reacting molecules in such a way that their reactive groups are brought close together. 2. In an enzyme-catalyzed reaction, more molecules have sufficient energy to react than in the absence of the enzyme. 3. Reactions catalyzed by enzymes take place at a lower temperature than they would without the enzyme. 36. What is the most appropriate set of controls to use in an investigation into the rate of an enzyme-catalyzed reaction over a range of temperatures from 25 OC to 65 OC? Substrate only at all temperatures tested. 37. When investigating the rate of reaction of the enzyme lipase on the hypothesis of triglycerides, the pH must be maintained at an optimum to prevent the lipase denaturing. What is the reason for this? The products of hydrolysis decreases the pH. 38. An investigation was carried out into the effect of an increasing concentration of substrate molecules on the rate of an enzyme-catalyzed reaction. All other variables were standardized. Which statement is correct? The rate increases to a maximum and then levels off. 39. Which statement about the effect of substrate concentration of the activity of an enzyme is correct? Above a certain concentration of substrate, an enzyme reaches its maximum rate of reaction. 40. Substances called furanocoumarins are found in some fruits, where they act as inhibitors of the enzyme CYP3A4. This enzyme is needed to break down toxins in the human digestive system, so dangerous levels of these toxins may develop if these fruits are eaten. What does this information suggest about molecules of the enzyme CYP3A4? They lower the activation energy of the toxin breakdown process. 41. The rate, enzyme-catalyzed reactions in human cells, is regulated. What may be involved in such regulation? 1. A change in enzyme concentration. 2. A change in substrate concentration. 3. Inhibition by the final product of the reaction. 42. The AIDS virus produces a long polypeptide that is hydrolyzed by a protease enzyme, producing several smaller peptides. This viral protease is the target of anti-AIDS drugs. Which feature is essential for the success of these drugs? A specific structure that inhibits only viral protease. 43. Which statement about the Michaelis-Menten constant, Km, is correct for an enzyme with a low affinity for its substrate? It has a high Km and reaches Vmax at a high substrate concentration. 44. The enzyme beta-galactosidase can catalyze the hydrolysis of 4 substrates with similar structures. Each substrate gives a different Km value. For which substrate does beta-galactosidase have the highest affinity? Substrate, 4 and Km mol Dm, 3, 1x 10 to 445. 2 enzymes are added to a solution containing a low concentration of a substrate that they can both use. Which statement is correct? The enzyme with the lower Vmax will use more of the substrate than the other enzyme. 46. An enzyme is modified for industrial use. It has a lower Michaelis-Menten constant, Km, than the unmodified enzyme. It has a higher affinity for its substrate. 47. The Michaelis-Menten constant for an enzyme-catalyzed reaction is referred to as Km, and the maximum velocity of such a reaction is known as Vmax. Which statement about of Km and Vmax is correct? An enzyme with a low value of Km has a high affinity to its substrate. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457776. 48. The Vmax and Km are determined for an enzyme-catalyzed reaction. What will be the effects on the Vmax and Km in the presence of a competitive inhibitor? Vmax stays the same, Km increases 49. The enzyme beta-galactosidase can catalyze the hydrolysis of 4 substrates, A, B, C and D, with similar structures. Each substrate has a different Km value. For which substrate does beta-galactosidase have the lowest affinity? Km equals 4x 10 to 3 moles Dm, 350. What is the definition of the Michaelis-Menten constant, Km, for an enzyme? The substrate concentration that gives half Vmax 51. Which is correct for an enzyme with a low Michaelis-Menten constant? Affinity of enzyme for substrate, high, substrate concentration at maximum reaction rate, low 52. How is the Michaelis-Menten constant, Km, is used? To compare the affinity of enzymes for their substrate 53. The painkiller ibuprofen reduces the activity of an enzyme involved in the production of prostaglandin. Ibuprofen binds reversibly to the active site of the enzyme. Which type of enzyme inhibition describes this example and why? Competitive inhibition, because ibuprofen and the substrate cannot bind at the same time. 54. Which effects can non-competitive inhibitors have on enzyme-controlled reactions? 1. Reduce the concentration of the product. 2. Reduce the rate of a metabolic pathway. 55. What affects the rate of an enzyme-catalyzed reaction when in the presence of a non-competitive inhibitor? 1. Enzyme concentration. 2. Inhibitor concentration. 3. Substrate concentration. 56. What would be the effect of adding a small amount of a non-competitive inhibitor of enzyme 2? Substance X would increase in concentration. 57. Which is correct for competitive inhibitors of enzymes? 1. They occupy the active site of an enzyme. 2. They can be used to control the rate of enzyme activity. 58. Which statement about the typical modes of action of a competitive inhibitor and a non-competitive inhibitor is correct? Competitive inhibitors will not alter Vmax, non-competitive inhibitors will reduce Vmax. 59. Aspirin inhibits an enzyme by reacting with an amino acid that forms an essential part of the 3D structure of the enzyme. Part of the aspirin molecule binds firmly with the amino acid. What describes this inhibition? Non-competitive inhibition. 60. Which features are correct for a competitive inhibitor of an enzyme-catalyzed reaction? Binds to active site, correct, changes shape of enzyme, incorrect, similar shape to substrate, correct, rate of reaction affected by concentration of inhibitor, correct? 61. Which statement about non-competitive enzyme inhibitors are correct? 1. The inhibitor can bind to the enzyme whether or not the enzyme has its substrate bound in the active site. 2. The inhibitor may bind at an allosteric site. 3. The inhibitor may not prevent substrate binding at the active site, but will prevent product formation. 62. Which statement correctly describes the action of competitive enzyme inhibitors? They limit the formation of enzyme-substrate complexes. 63. Tyrosinase is an enzyme that catalyses the concentration of the amino acid tyrosine into the black pigment melanin. It is responsible for the black fur color of some rabbits. A group of rabbits kept at 30 OC resulted in 90% of the rabbits with light fur color. A second group of rabbits kept at 10 OC resulted in 90% of the rabbits with black fur color. Lecture notes for AS Level Biology by Ahmed Rashid. Chat the cell number 03004610457777. Which hypothesis is supported by these results? An inhibitor is present in rabbit skin cells that can bind strongly to tyrosinase when the external temperature is 30 OC. 64. Which statements about enzyme inhibitors are correct? 1. Competitive inhibitors may be similar shapes to the substrate. 2. Competitive inhibitors bind to the active site. Non-competitive inhibitors alter the shape of the enzyme. 65. Some inhibitors of enzyme reactions bind to the enzyme-substrate complex. Which statements about this type of inhibitions are correct? The inhibitor is non-competitive. 2. The initial rate of reaction is reduced. 66. Which levels of protein structure are always involved when competitive and non-competitive inhibitors bind to enzymes? Competitive, tertiary, non-competitive, tertiary. 67. Which statements about the effect of all enzyme inhibitors are correct? Reduce the rate of the enzyme-catalyzed reaction. 68. Which is correct for a competitive inhibitor of an enzyme? Inhibitor binds to the active site of the enzyme and increasing the substrate concentration decreases the effect of the inhibitor. 69. Which statements about competitive inhibitors of enzyme action are correct? 1. Increasing the concentration of the enzyme substrate will reduce their effect. 2. They bind to an enzyme at its active site. 70. How does increasing concentration affect the rate of an enzyme-catalyzed reaction in the presence of a competitive inhibitor? The rate of the reaction increases. 71. The rate of enzyme-catalyzed reaction in human cells is regulated. Which may be involved in such regulation? 1. A change in enzyme concentration. 2. A change in substrate concentration. 3. Inhibition by the final product of the reaction. 72. The equation shows a reversible reaction. In this reaction, on which molecule does an active site occur and what types of reaction occur at 1 and 2? Active site present on sucrase, reaction at 1, hydrolysis, reaction at 2, condensation. 73. Following a heart attack, the enzyme lactate dehydrogenase leaks into the blood plasma from damaged heart muscle. Which steps are required to obtain the best estimate of lactate dehydrogenase activity in a sample of blood plasma? Incubation with substrate for lactate dehydrogenase required and sterilization of blood plasma by heating and incubation with lactate dehydrogenase inhibitors are not required. 74. During the production of apple juice, enzymes are used to break down the components of the cell walls. Which carbohydrate will be produced by this hydrolysis? Beta-glucose. 75. Which properties are characteristic of a non-competitive inhibitor of an enzyme? Binding, not at active site, effect of adding more substrate, does not reduce inhibition. 76. Which statement is true of all enzymes? They reduce the amount of energy required to start a reaction. 77. What is the effect of increasing substrate concentration on the degree of inhibition of an enzyme-controlled reaction? Competitive inhibition, decreased; non-competitive inhibition, increased. 78. During the development of AIDS virus, a long polypeptide is hydrolyzed by a protease enzyme, producing several smaller peptides. This viral enzyme is the target of new anti-AIDS drugs. Which feature is essential for the success of these drugs? A specific substrate that inhibits only viral protease. Notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457778. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457779. 4. Cell membranes and transport. Cell membrane is a phospholipid bilayer that regulates